Welcome to the deep dive. Today, we're digging into something really fascinating. It involves electroconvulsive therapy, ECT, and some surprising effects it has on the brain.
Right. It's often seen as quite intense, but the outcomes can be significant.
Exactly. And we've got a scientific paper here that uses optogenetics in mice to sort of mimic ECT-like stimulation.
A really clever approach to get at the underlying mechanisms.
Yeah, so prepare to explore how this powerful intervention might actually trigger beneficial changes deep inside brain cells.
The central question is really quite counterintuitive, isn't it? How can such strong stimulation be good for the brain?
That's the mission for this deep dive. This research suggests it might kickstart a kind of cellular reprogramming, maybe even pushing cells back towards a younger, more immature state.
Which is fascinating because, normally we think of really high-intensity brain activity as damaging. It can cause cell death, loss of connections.
Right, synapse loss, hyperexcitability,
Hmm.
generally bad news.
But then you have ECT, which involves significant neural activity, and yet it's known to be effective for certain conditions, particularly severe depression.
Okay, so let's unpack that paradox. We know ECT can work, and the research shows real, tangible changes, neuroplasticity, right? The brain rewiring itself.
hmm Things like neurogenesis, neurons growing in the dentate gyrus, and even the hippocampus getting slightly larger.
Crucial areas for memory, learning, but the how, that's been the mystery.
Precisely. Despite knowing it works, the cellular nuts and bolts of ECT and even related therapies like TMS or TDCS have been pretty unclear.
It's like knowing the light switch turns on the bulb, but having no idea about the wiring inside the wall.
That's a perfect way to put it.
Oh.
And that's the gap this study aimed to fill. They wanted to see exactly what happens inside the cells.
So how did they do that? You mentioned optogenetics?
Yeah, they used a technique they call REPO-PS, that's repetitive optogenetic stimulation. Basically, they used mice where specific neurons, the granule cells in the dentate gyrus.
Which is a key area for ECT's effects.
Exactly. These neurons were engineered to have light-sensitive switches, so the researchers could turn them on very precisely using light.
Ah, so much more targeted than, say, older methods like seizure-inducing drugs or just general electrical stimulation in animals.
Absolutely. Those older methods like animal ECS lack that fine control. With REPO-PS, they could deliver really specific pulses. Think short bursts, just 10 milliseconds.
I mean, bursts.
Yeah, repeated 10 times a second for 5 minutes each day. And then they did this daily routine for either 3 days or 10 days.
To see if the duration made a difference.
Exactly, to see how the length of this precise stimulation affected the outcome.
Okay, so they've got this very controlled ECT-like stimulation. What did they actually see happening in those brain cells? The really big finding seems to be this idea of cellular dematuration. What exactly is that?
Right, so what they found, especially after the longer 10-day stimulation period, was that these dentate gyrus cells started to, resemble younger, more immature cells.
Longer.
Yeah, it's like they took a temporary controlled step backward in their development. The idea is that this strong, precise stimulation might be making them act younger, more flexible again.
A step back. That sounds so odd. Usually we think of cells maturing, specializing. How did they even measure this dematuration?
Good question. They looked at a few key things. First, a protein called calbindin. Its levels usually go up as these granule cells mature.
Okay.
Well, after APOPS, especially the 10-day round, calbindin levels went down and stayed down for a while. It was more temporary after just 3 days.
Interesting. And they figured out why.
They found that calcium rushing into the cells during the stimulation was crucial for that calbindin decrease. So light triggers activity, calcium comes in, and that affects the maturity markers.
Okay, so one protein marker changes. What else pointed to this cellular youth potion effect?
Well, this is maybe even more striking. They looked at the overall pattern of gene activity, which genes were turned on or off in these stimulated cells after 10 days. And they found that this pattern looked remarkably similar to the gene activity patterns found in the brains of very, very young, like infant mice.
Wow. Seriously, the adult cells started expressing genes like infant cells.
Using a statistical test, they found a really high degree of similarity. That wasn't the case after just 3 days of stimulation. It really suggests a shift back towards an infant-like genetic profile.
That is pretty mind-blowing. And crucially, this wasn't just in mice, right? They connected this to humans undergoing ECT.
Yes, and that's vital. They looked at gene expression in postmortem dentate gyrus samples from human patients who had received ECT versus those who hadn't.
And found similar patterns.
Found evidence of similar immature-like gene expression patterns in the ECT group, It really strengthens the idea that what they saw in mice might be relevant to humans.
Okay.
And there was more. Both the 10-day Repop-ES mice and the human ECT patients showed changes in genes related to synapses, the connections between neurons.
No.
They saw lower levels of genes, like GRIA1 and GRIA2, think of them like input receptors and also CMK2 genes, but higher levels of a gene called ARC, which is involved in changing synapse strength.
So the connections themselves were shifting towards a more immature, maybe more adaptable state.
That seems to be the implication. It paints a picture of quite significant cellular reprogramming, affecting not just the cell's internal state, but also how it communicates.
And this reprogramming went even deeper than just gene activity levels, didn't it? Something about the genome structure.
That's right. They used a technique called ATAC-seq to look at chromatin accessibility, basically, how tightly the DNA is packed up.
Which affects which genes can be easily read.
Exactly. And they found that Repop-ES caused long-lasting changes in this chromatin structure, making different parts of the genome more or less accessible, so that the stimulation was changing the very landscape of the genome.
A really fundamental change. Okay, we're seeing these profound shifts at the cellular, genetic, even genomic level, but does it do anything? What about the mice's behavior? Did these changes translate?
They did. And this is where it really connects to the potential therapeutic effects. After 10 days of Repop-ES, the mice showed behaviors reminiscent of ECT effects.
Like what?
Increased movement, more locomotor activity, both right after stimulation and also sustained over the longer term.
Okay.
And perhaps more importantly, they showed less depression-like behavior. In standard tests like the tail suspension or forced swim test, they spent significantly less time immobile, less time showing signs of giving up.
And this only happened with the longer stimulation.
Right. These behavioral improvements weren't really seen with just 3 days of Repop-ES. It lines up nicely with the more sustained cellular changes in the 10-day group. The longer stimulation seems key.
It's amazing how these tiny cellular shifts can ripple out into observable behavior, but the brain processes information. Did Repop-ES change how these neurons were actually, coding things?
Yes, they looked at that too, using calcium imaging while the mice were actually moving around.
So they could watch the neurons fire in real time.
Essentially, yes. And they found Repop-ES actually increased the number of speed cells, neurons, that fire based on how fast the animal is moving. And it made their speed coding more accurate.
Huh. Maybe that explains the increased movement.
Could be part of it. But there was a flip side. Repop-ES also seemed to interfere with how well the dentate gyrus formed stable spatial maps, how the mice learned to navigate space over time.
So maybe a trade-off. Better speed coding, but worse spatial mapping. That sounds like it could relate to potential side effects, maybe memory issues reported with ECT.
It's certainly a possibility. It suggests the effects are complex. Now, perhaps the most unexpected finding was this link to the cell cycle.
Right, you mentioned that mature neurons don't usually divide, so what's going on there?
This was really novel. They found that the repeated stimulation triggered the activation of genes normally associated with the G2 and M phases of the cell cycle, the preparation and execution stages of cell division.
In mature neurons?
In mature neurons. And this genetic program stayed active for over 2 weeks after the stimulation stopped.
That sounds wrong, like a biological short circuit. What was the evidence?
Well, they saw higher levels of key cell cycle genes, cyclin A2, cyclin B1, B2, CDC25C, and they saw physical changes in the nucleus. The nuclear lamina, a support structure, got disrupted. There was more of a specific marker called phosphorylated histone H3, which is a hallmark of mitosis.
Cell division.
Right. And the chromatin looked more condensed, also typical of dividing cells. And crucially, this wasn't just in newborn neurons. It was happening in the established, mature granule cells.
But wait, you hear about aberrant cell cycle reentry in things like Alzheimer's or epilepsy, and it's usually bad news leading to cell death. How is this different?
That's the critical point. Unlike in those disease states, the Repop-ES stimulation didn't cause significant cell death. The nuclear changes were also maybe less extreme than in full-blown division.
So it's like the cells started down the path to division, but then stopped.
Exactly. It seems to induce an arrested state, maybe somewhere between G2 and early M phase, like revving the engine but keeping the brakes on.
Okay, still very weird, but maybe not necessarily destructive. And the study singled out one molecule, cyclin B, as being really important here.
Yes. Cyclin B is a master switch for entering the M phase. So they tested its role using mice genetically engineered to lack cyclin B in those neurons.
And what happened when they gave those mice Repop-ES?
Many of the effects were blunted. The long-term drop in calbindin, that maturity marker, was reduced. The increase in that mitosis marker, pH3, was less. The chromatin condensation was less. And significantly, the long-lasting increase in movement didn't happen.
Wow. So cyclin B is really mediating a lot of this.
It seems to be a crucial link connecting that repeated neuronal firing to this immature-like state and the behavioral changes. It acts like a central hub in this whole process.
Okay, so cyclin B is a key player, and there was another molecule involved in the longer-term changes, delta, FOSB.
FOSB, yes. It's a transcription factor, meaning it controls other genes. They found that the activity of cyclin B, along with its partner enzyme CDK1, leads to increased levels of OSB.
And FOSB then drives the lasting changes.
It appears to mediate that persistent genomic remodeling, the chromatin changes we talked about. They also found that higher FOSB levels correlated with lower calbindin levels, fitting with this idea of pseudo-immaturity.
I see.
And interestingly, while the initial quick spike in FOSB after stimulation didn't need cyclin B, keeping those levels high over the long term did require cyclin B.
So it's like a cascade. Stimulation past cyclin B pathway sustained HOSB-FOSB long-term genomic and cellular changes.
That seems to be the picture emerging from this research. It's complex, but it connects the dots from stimulation to these very fundamental changes.
So pulling this all together, what are the big implications here?
Well, it offers a potential cellular mechanism for why ECT might work. It suggests it induces this reversible immature-like state.
And that state might be therapeutic.
The idea is that this temporary reset could restore flexibility to circuits that have become rigid or dysfunctional in conditions like depression. It might reset metabolic and synaptic balance.
But it also potentially explains some downsides.
Right. Things like memory issues could be linked to those long-term changes in synapse genes or the altered neural coding, like the spatial map disruption they observed.
Yeah.
They did find some deficits in remote memory in the mice.
So it's clearly a double-edged sword. Therapeutic potential, but also risks. Did they compare this to other treatments?
They did. They noted similarities between the REPAPS effects and effects seen with both traditional animal ECS and also SSRI antidepressants.
Suggesting a common pathway?
Possibly a common theme, yeah. Maybe different treatments converge on reactivating some form of plasticity, nudging neurons towards a more adaptable state.
And this immature state itself, it could be good or bad, depending on the context.
That seems plausible. Therapeutic at these controlled stimulation levels, maybe, but potentially contributing to disease if the stimulation is excessive or uncontrolled, like a bolstable switch.
This really feels like it opens up new possibilities for refining these kinds of therapies.
Absolutely. Understanding this cellular reprogramming process could lead to more targeted brain stimulation techniques, maybe designed to maximize the benefits while minimizing side effects.
So let's wrap up this deep dive. We've seen how ECT-like stimulation, mimicked precisely with optogenetics in mice, seems to push mature brain cells into a temporary younger-like state.
A state characterized by widespread changes in gene activity, even the structure of the genome itself, and this really surprising engagement of cell cycle machinery.
And this cascade leads to both positive behavioral changes, like reduced depression-like behavior, but also potential negatives, like effects on memory processing.
And it brings us back to that core surprising idea that inducing a form of cellular immaturity might actually be beneficial. It definitely challenges our standard view of brain maturation.
It really does. So here's a final thought to leave you with. If this research reveals such a fundamental way cells respond to stimulation by becoming more plastic or immature, how else could we potentially harness this? Could we fine-tune stimulation, maybe target specific cell types even more precisely to get the therapeutic effects without the memory issues? Could this lead to entirely new ways to treat complex neuropsychiatric disorders? Definitely food for thought. Thanks for joining us.